Alright, let's dive into something that's honestly mind-blowing, and no, I'm not exaggerating. Imagine nanoparticles, these tiny, almost invisible structures, acting like stealth agents to sneak antibodies into cells. Sounds like sci-fi, right? But it's real, and it's happening right now. Researchers have developed a delivery system using lipid nanoparticles that can smuggle therapeutic antibodies into cells, potentially revolutionizing how we treat diseases like cancer, inflammation, and even Parkinson's. Personally, I think this is one of those breakthroughs that doesn't just push the boundaries of science, it redefines them. What makes this really interesting is the sheer ingenuity behind it. The team, led by Ismail Alamgir and involving experts like Chris Alabi and Matt DeLisa, figured out how to cloak proteins with a negatively charged ion. This allows them to hitch a ride on positively charged lipid nanoparticles, essentially tiny fat bubbles. Once inside the cell, the proteins uncloak and do their job. It's like a molecular heist, but instead of stealing something, they're delivering life-saving therapies. What many people don't realize is how challenging it is to get antibodies into cells in the first place. These molecules are huge, 150 kilodaltons, and they can't just waltz through the cell membrane. This technology changes the game entirely. In my opinion, one of the most fascinating aspects is the collaboration that made this possible. The Cornell team developed the cloaking technology, and then researchers from the Technion-Israel Institute of Technology applied it to target Parkinson's disease. This wasn't just a lab experiment, it was a global effort. Alabi mentioned how satisfying it was to see their technology work in a completely different lab, under different conditions. If you take a step back and think about it, this is a huge leap toward clinical translation. It's not just about proving a concept, it's about showing that this can work in real-world scenarios. A detail I find fascinating is the versatility of this approach. They didn't just stop at Parkinson's. The Cornell team also demonstrated its effectiveness in treating lung inflammation in a mouse model. This raises a deeper question: if this works for such diverse conditions, what else could it be applied to? Cancer, autoimmune diseases, maybe even more neurological disorders? The possibilities are staggering. What this really suggests is that we're just scratching the surface of what lipid nanoparticles and cloaked antibodies can do. From my perspective, the launch of Cloak Bio by Alabi, DeLisa, and Alamgir is a massive vote of confidence in this technology. They're not just publishing papers, they're building a company to take this to the next level. This isn't just about academic curiosity, it's about making a tangible impact on healthcare. Personally, I'm excited to see how this evolves. Will we see FDA-approved therapies in the next decade? Could this be the key to treating diseases that are currently untouchable? These are the questions that keep me up at night, in a good way. If you take a step back and think about it, this research is a perfect example of how interdisciplinary collaboration and innovative thinking can solve some of the most stubborn problems in medicine. It's not just about the science, it's about the potential to change lives. So, here's my closing thought: what if, in the future, we look back at this moment as the turning point in how we treat diseases? What if these tiny nanoparticles become the heroes of modern medicine? Let me know what you think in the comments. I'd love to hear your take on this.